If everyone would like to come in and take a seat, we are going to get rolling in just a minute or two. So welcome to everyone who is joining us online as well as in person here today. So this is day 2 of the Entomological Collections Network meeting. And we're starting off the day with another symposium, Collections of the World. And this is the 4th year running, and it's really a great way to highlight the success of the virtual and hybrid meetings that we have going. We have collections tours from numerous countries around the world. Unfortunately, we've had 3 cancellations, but the good news is we do have another talk that will be filling in one of those gaps. So just a reminder that the code of conduct still applies to those online and those in the room. If you can mute your phones if you're here, that would be fantastic too. The way that this session is going to run, we are going to play the videos of these collections tours, and then there'll be time for asking questions. So the people that are in person can come up to the podium where we'll ask them whatever we would like. And for those online, we ask you to unmute and we can ask people those questions too. So that did not take 5 minutes for that introduction. But yeah, we might kick it off. So the first tour is that of the Hong Kong Biodiversity Museum. And Alina is joining us online.
My name is Aline. I'm the curation manager for our museum. And today we're talking about the Hong Kong Biodiversity Museum, developing natural history and education in a concrete jungle. So this concrete jungle is Hong Kong. It's located between the Tropic of Cancer and the equator line. So it's on the edge of the tropical region, but it's still in the tropical region, and it's located in the south of China. So Hong Kong has this big portion, the continental portion, and then 2 larger islands and hundreds of smaller islands. And in Hong Kong, you can find these 2 completely different landscapes. The urban area with a lot of buildings, skyscrapers, and then the natural area with forest, mountains, a lot of different environments, and many species. It's estimated that Hong Kong has about 30,000 species. It's impressive given the size of Hong Kong, but probably we have much more species than that. And in the Hong Kong Island, there is the University of Hong Kong, which hired Professor Benoit Guénard in 2014. And Professor Benoit, he has this passion for education and science and for biodiversity. So he wanted to promote more biodiversity in Hong Kong. And he came across this big collection that nobody was curating in HKU, and then he started putting everything together because the specimens were quite spread in the building, in different rooms, in different boxes, different cabinets, and then he put everything together. He applied for a grant, and finally in November 2020 we founded the Hong Kong Biodiversity Museum. So far, we have about 42,000 specimens in our museum. I will not talk about each of the groups. You guys can check that later. And in numbers of species, we have about 2,300 species that are identified. But probably we have much more because most of the insects and invertebrates, they are not identified at species level. They are identified at family, order. So if we identify everything, we'll probably have much more species than that. We also have a plant collection with about 7,000 specimens and 2,000 species. So it's the same thing. We are still working on it, and probably we have much more, at least double. number of species. So after we start working with the collection, organizing everything, databasing everything, knowing the number of species, number of specimens, what species we have, we finally were able to open it for the public. So we opened on the 22nd of May 2021, and we choose this specific day because this is the day, International Day of Biodiversity, of biological diversity. So we open 3 times per week on Wednesdays, Saturdays, and Sundays, and we have sections 45 minutes long, and it's short because we want more people to come to see the museum, and then they can come back again and bring family and friends, and also because we have our sections are guided, so we have 2 guides for each section, because we like to talk to people, to explain about biodiversity. We are very people-oriented, and we really like to interact with everyone that comes to visit our museum. So far, we have had 26,000 visitors, and we have these regular visitors during Wednesdays, Saturdays, and Sundays, but we also have some private groups that are people that come in other days, and they come usually from schools and NGOs that we collaborate with. We are also on social medias, on Facebook, Instagram, and Twitter, and in total we have more than 10,000 followers on social medias. And since we opened 2 years and a half ago, every week we post something about one of the species in our museum. So we showcase our collection every week, and that's very nice because we have people that read about one specific species that we posted that week on social media, and then they come to visit our museum and they remember some interesting facts about that species, and they talk about it very enthusiastically. So it's really, yeah, it's a very interesting way to showcase the collection virtually and in person, right? We also, as I mentioned, we have some collaboration with NGOs and schools, and this is one NGO in Hong Kong called Blind Union, and we have had this visit where blind people came to visit us, and usually blind people, they don't have access to museums, right? Because in museums they cannot touch and they cannot see what are in the exhibitions, but we selected some pieces in our exhibition that they could touch, and also the Blind Union, they provide this notebook with a texture, so the blind people could also feel the different texture and understand better the structures on the specimens. It was a very rewarding collaboration. We also have external workshops, and we try to show people biodiversity in a different way. We bring microscopes, we teach them how we do science, how we work with the specimens, and especially talking to children about it. It's very inspiring. We also have the research part. So we have collaboration with PhD students from different universities, from researchers from HKU and from other places. And we also serve as a depository institution for taxonomic studies. And also we do research in the museum in collaboration with other lab members. We also provide training to the community, especially for the university community, HKU and other universities. And so far we have trained 50 interns and 20 volunteers that come to us to learn about biodiversity and spread the word of biodiversity to others. So it's been very rewarding working in this museum and talking about biodiversity to so many people. And I don't have more time to talk about it, so I'd like just to thank everyone for watching this presentation, and especially ECN for providing this space. Thank you so much.
Thank you so much. There is room for about 5 minutes of questions. So if anyone in the room or online has a question for Aline, and she is online currently. Yeah.
It seems like people only see the museum through guided tours, but there's no sort of like this non-guided access. Is that true? I'm curious about that.
So the question was, it seems like the museum, you can only see it through the guided tours. Is there any way that the visitors can also do it non-guided? I think Aline is online. Are you able to unmute there? So we can see her online, but unfortunately, we are having technical issues. But perhaps there might be an opportunity for her to answer that in the chat. We'll keep an eye out on that. So they're only so the answer online is that they are only able to do guided tours so far, but hopefully it's something that they'll be able to do in the future. There are any other questions in the room or online? Jennifer?
They have a dedicated insect collection, and if that's going to be database or digitized somehow.
So the question is, do you have a dedicated entomology collection, and is it going to be digitized? And she also is apologizing because her microphone doesn't seem to be working, but there looks like she is typing. They are currently working on the process of digitization right now. So we look forward to seeing that in the future. Okay, so next up, we have another tour from the Asian region, and this time it's the insect collection of Seoul National University, South Korea. And this video was prepared by Lee.
Hi, I am Seung-Yeon Lee, working on beetles. I am a graduate of Seoul National University, and I'm excited to take you on a virtual tour of our collection. So let's begin. Seoul National University sits on the other side of the world from where we are now, National Harbor, Washington. It's a 15-hour flight from here to Seoul. Now we are in Seoul, and you'll find Seoul National University down south in Seoul. It's the largest university in South Korea. And SNU insect collection is located in the middle of the campus. So let's take a trip to Seoul National University together. Here's the main gate of Seoul National University. The campus is nested at the base of a mountain, so I usually hop on a bus to get around. College of Agriculture and Life Science is just about a 5-minute bus ride from the main gate. There are several entrances, but this here is the main one. The 6th floor of this building houses the entomology department. Inside, you will find the insect biosystematics lab and the Seoul National University insect collection that we're going to check out today is right next door. Seoul National University boasts Korea's largest university-affiliated insect collection, home to roughly 0.7 million specimens. Out of these, 370,000 are dried specimens, 280,000 specimens are stored in fluid, and the remaining 50,000 are slide mounted. You might think this isn't much for a nation's biggest collection, but you see, the collection doesn't have a long history as other collections. 1900s, South Korea was one of the poorest countries in the world. The Korean War in 1950s left the nation in ruins, still one of the poorest countries. Thankfully, since the 70s, the country has been on a fast track of economic growth. So, South Korea has only recently begun to show interest in insect diversity and taxonomy. Seoul National University was called Suwon Higher Agricultural School during the Japanese colonization era in the early 1900s. It was renamed Seoul National University in 1946 and moved to its current location in Seoul in 2003. So, we have some pretty old specimens, some dating back to the 1930s. For example, we've got specimens of the fall webworm outbreak in 1960s, the spotted lanternfly outbreak in the early 2000s, and the recent emergence of exotic cerambycids. These historical collections are valuable because there are very few old specimens in Korean collections. Of course, as a university-affiliated institution, we also have specimens for educational purposes. We use them in undergraduate and graduate courses on insect taxonomy. Plus, we get visitors as part of our public outreach. Most of the specimens are from South Korea, with a mix of small portion of Southeast Asian specimens. Since the 2010, we've signed MOUs with countries in Southeast Asia, allowing us to collect specimens from that region. Our Southeast Asian samples come from Myanmar, Laos, and Cambodia. And since the collection is a part of Seoul National University, lots of PhD alumni and current students have collected various insects. Their research interests encompass Hemiptera, Hymenopteran, Coleoptera, and Lepidoptera. Here's the myriad collection. The Southeast Asian longhorn beetles and some Trichteratomidae. This is the psyllid collection. Some of our psyllid specimens are stored dry, while others are preserved as a slide mount specimens. This is where the slide specimens were stored. It's on one side of the wall of our lab. This collection mostly contains insects like psyllids, aphid, scale insects, and a few ticks and mites. Now the real deal of our collection is the fluid specimens. We've got 5 freezers packed with samples. In our lab, we do molecular phylogenetics and some population genetics on various groups. And the DNA grade samples used in these studies are stored in ethanol and kept frozen. It's all organized with collection numbers, and it's one of the best maintained parts of our collection. So that's the insect collection at Seoul National University. If you like to visit or have any questions, feel free to drop our PI an email at the address below. And if you want more information, visit our website. Thank you.
We have time for probably 2 or 3 questions.
Yes.
When you chose the drawers, there's a lot of specimens with like red labels on the bottom. Was that were that holotype types or a different way to label?
Actually, different people like maintain their collection. For example, some Hemitarn people, they like maintain their collection. So I'm not sure about that, but surely we have many types. So I think that, yeah, with red cards will be type specimens. Yeah, I think so. Oh, yes.
Is there a history of amateur insect collecting in South Korea?
Oh, yes, of course, because like I was a beetle enthusiast actually. Yeah, so yeah, there are so many enthusiasts. So what do you want to see here
Like,
in specific?
Do many of the insect specimens that you have come from academic collectors or amateurs?
I think that's mostly from academic collectors. Like amateurs has their own collection in their home. For example, I have my collection in my homes. Yeah, that is what it is. Yes.
Well, you started receiving big collections from amateurs when they pass away and they give them to the museum.
I think they don't give us samples. We have another institution that we call it National Institute of Biological Resources or something, and most of the donated specimens go to that collection. That's near our collection. It's like 1.5 hour drive. Yes. Yes.
Do you not have earthquakes there? I live in a very earthquake.
Oh.
State. And when I saw that beautiful slide collection, I worried about.
Okay. I actually we don't have earthquake. Like yeah, I think Japan has like serious earthquake, but not in South Korea. But yeah, that's like kind of I think it's fragile, so we need to think about how to make it safe. Yes.
Well, excellent. Oh, we have one more.
So these samples that you have stored in your freezers, that's miscellaneous things, and is it prepared for, or at least thought of for DNA analysis?
Yes. Yeah. So we collect the live samples and then put them directly into the ethanol. And for example, some are large, like I study cerambicids, and there's some of them are large, right? So we put just one legs or some muscles into the tube. Yeah. So all of them are for molecular things. Yes.
There is a question online. Is there someone that is currently working on the scale insect collection there?
Oh, yes. But he's now in Japan, in Okinawa. Yes, he's doing postdoc there, but his collection is quite good, I think. Yeah, I think he has many like Southeast Asian cocoids. Yeah. So, oh yeah.
I like your technique of showing the collection drawer and pointing out and having photographs superimposed on that. Yeah, you could actually see investments better. That's very nice.
Well, thank you very much. It's an absolute pleasure getting to see all of these collections around the world that most of us may not have known about or got to see. So next up, we have a tour of the Lepidoptera collections at the Natural History Museum, London, presented by Jeff Martin.
Hello, I'm Jeff Martin, Principal Curator of Lepidoptera, the National History Museum, London, UK. I'm now going to give you a whistle-stop tour of our Lepidoptera collections. First of all, who works on the collection? We have a team of 7 curators as follows. Blanca Huertas is in overall charge of the Lepidoptera collections and responsible for all the Papilionoidea except the British and Irish. I'm responsible for the Geometroidea, Draconoidea, and the British and Irish collections. Alberto Zilli is responsible for the Noctuoidea. David Lees is responsible for the Microlepidoptera. Alessandro Giusti is responsible for the rest of the Macrolepidoptera. Both Richard O'Brien and Robin Crowther are working on Papilionoidea. We also have several scientific associates who also work on the collection in an unpaid capacity. I won't go through all their names, but there should be some familiar faces if you work on Lepidoptera. Their role was very important, as much of the taxonomic work done on the collection is produced by our scientific associates. As you can see, we have more scientific associates than staff. We also have a team of around 10 volunteers that help with the day-to-day curation of the collection. Now onto the buildings and important dates and the ever-changing acronyms. The museum opened as the British Museum in 1753 in Bloomsbury, London, with the acronym still used by many as the BM. The Natural History Collections outgrew this site, and in 1881, all the Natural History collections were moved by stagecoach at night to the current site in South Kensington, and the acronym became BMH. In 1992, the museum was renamed as the Natural History Museum, acronym became NHM. In 2009, the Darwin Centre opened, which now houses the Lepidoptera collection over 4 floors with proper environmental controls for the first time in the collection's history. The acronym is now NHM UK. Some collections metrics. We have 13.5 million specimens housed in 80,000 drawers. There are 125,000 primary type specimens. We certainly have the best global coverage, with strengths being vast numbers of specimens from Africa, India, Southeast Asia and Australasia, but significant gaps in particular from both Americas and the Eastern Palearctic. However, there is a lack of recent material from many areas. There is a separate British and Irish collection of around 1 million specimens. There is also a small spirit collection of mainly larvae. The most important part of the collection is the type material, of which there are 125,000 primary type specimens. This is likely to be an underestimate. Most types are contained within the main series of specimens of a given species. Recently, we have begun to separate type specimens from the main series, as this makes access and auditing considerably easier, ensures the safety of the specimens, and speeds up the digitization of the specimens. Separate type of collections so far are the Madagascar material, the birdwing butterflies, recent acquisitions, and material currently undergoing recuration. A word of caution here concerning the round BM type labels. We have recently found that many are inaccurate, and large numbers of type specimens, particularly long syntypic series, bear no round type label at all. Original description should always be checked. Here we have a slide showing 10 authors that have described more than 1,000 taxa now housed in the museum. Butler was employed by the museum from 1863 to 1901 and described taxa across all groups. Herbert Druce's collection came in via Godman and Salvin and the Joycey collections and contain hundreds of taxa described by him. I was surprisingly unable to find an image of Druce. A large part of the Frühstorfer collection was purchased in 1937, some 52,000 specimens and very type-rich. Hampson was employed by the museum from 1895 to 1913, producing 16 volumes of his catalogue of the Lepidoptera Fellaini in the British Museum and described thousands of taxa. The Merritt collection of 100,000 Microlepidoptera was bequeathed in 1938, containing thousands of types. Moore described hundreds of taxa from the Indian subcontinent. Prout worked on the Geometridae and described thousands of taxa. Walker was contracted by the museum to produce the 35-part list of the specimens of Lepidopterus insects in the collection of the British Museum between 1854 and 1866. Here many thousands of taxa were described. Wassington donated a collection of 260,000 specimens in 1910, which alongside the hundreds of taxa described by him also contained type material described by Zeller. Warren was another prolific describer of the Geometridae, but again, I was unable to find an image of Warren. On this slide, we come to the Rothschild collection, which was acquired in 1939 and was the largest bequest the museum has ever had, with some 2.25 million specimens, and contains over 1,000 taxa described by Rothschild. Other type-rich collections acquired around the same time include the Joycey bequest in 1934, more than 300,000 specimens, and the Overture collection of 750,000 specimens, which was purchased in 1927. The Overture collection also contained type material from other authors such as Bordeval and Guéné. In more recent times, other important acquisitions include the Wiltshire collection donated in 1979, with 30,000 specimens and 400 types, and the Inui collection donated in 1992 with 200,000 specimens and 700 types. The last major type-rich collection, the Kaju collection, was purchased in 2008 and contains 250,000 specimens and 200 types. Here are 4 type specimens. As you can see, the quality of the actual type specimen is quite variable. The first specimen is the type of Queen Alexandra's birdwing, which is in poor condition due to being shot rather than collected with the usual butterfly net. Next up are typical Hampson Walker type specimens. Note the fact that both sides of the label have information. And finally, an extreme example of an individual specimen actually being the type of 2 different names described by 3 authors. Collections access. Having a large number of type and other specimens under our care means we have an international responsibility to ensure access to these specimens. This is achieved through visits to the collection, physical loans, and increasingly digital loans. In an average year, 140 visitors over 600 visitor days visit the collection. Some 10,000 specimens are on loan at any given time, and increasingly access is given via digital loans. That is images of specimens, preparations, and labels. On the left, we see specimens box ready to go out on a physical loan. On the right, examples of images sent as a digital loan. Recuration is essential for improving access. For example, the geometry day completed by John Cheney, now retired, in 2016, is almost a self-service collection with very little input needed from the curator. Other parts of the collection are not sorted, amalgamated, or have not been updated for 50 years or more. Part of our job responsibility is to amalgamate various collections into one coherent series. Only then can the visiting researcher be sure they have all the material they need. Also, digitization of Celtic collections just cannot be done. Digitization continues at pace with some 800,000 specimens currently available in the NHM data portal, with more specimens being added daily. We're still acquiring material. We receive offers of collections on an almost weekly basis, and there are some surprisingly good collections still in private hands, such as the McCleary collection of African butterflies, seen here on the left. Many visitors to the collection also donate material. There are funds to purchase material, with a recent purchase being the Vinci Guerra collection of Cassinidae, Uranidae, Saturnidae, and Abramidae, seen here on the right. And of course, field work is still carried out by both staff and scientific associates. And finally, visitors are always welcome. We have thousands of drawers of unsorted material that need working up. I will leave you just another reminder of the scale of the collection. Here are images from the 4 floors. There are 2 corridors of cabinets like this per floor. Thank you for watching, and any questions?
Yeah, I'm curious about your digital loans. You know, a lot of our digital data gets used. You don't know who used it sometimes, but it sounds like you keep track in a loan system of digital data use.
Yeah, somebody, I mean, obviously stuff that's digitized is digitized by a different team, but obviously if you want to look at something in particular, you can send a request in. I want to look at specimens of X, Y, and Z, and they'll be photographed for you on your behalf. The copyright will be a Creative Commons, so although you'll have the images, anybody can use them and they'll get uploaded to the data portal in the same way. But it's a request for specimens, and obviously Lepidoptera being nice and flat, that works out quite well. Obviously stuff like more rounded insects, it's not quite so easy. Hi.
I guess my question is elaborating on Garrett's question. Can you keep track of those in the database as a loan?
Yeah, obviously we have to create a specimen record in our database. The photographs taken by somebody else, and then this is uploaded to that data portal I showed on the previous slide, so it will become publicly available.
Is it treated as a transaction, like loan numbers such as?
Yeah, it gets a loan number. In our system, you can have a loan number for a physical loan or a digital loan, so it is tracked.
Well, perfect.
No,
Thank
thanks.
you very much. So the next 2 collections tours, one from Santo Domingo, the Dominican Republic, and also Vienna, Austria, they have unfortunately been cancelled. But the good news is we do have a talk And this one is on the collection of ants at the Guwahati University, Assam, India. And Bulbini Kadakor is here to present.
Very good morning to all of you. Myself, Dr. Bulbini Kadakor, representing Guwahati University, Assam, India. Going to speak about the collection of ants present in Guwahati University, especially in Assam part. So ants, we all know, they're fascinating creatures that attract human attention for different reasons, for their disciplined movement, for their selfless services to their colony members. Many diverse roles they play to the ecosystem, like decomposers, pollination service, nutrient recycling, many more, seed dispersal, etc. But clinging cells, insects are declining very rapid pace, and ants are also not an exception. So, it's not going on. It's not coming. Okay. 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 So globally, there are 16,651 valid ant species. Approximately 845 species are reported from India. And Indian endemicity of ants is 31%. I'm representing Assam, which is a part of Eastern Himalaya Biodiversity Hotspot region. And before 50 to 70 years back, there are reports of 2-1 species presence in that particular regions. It was reported in a checklist, Bharti et al. But there is a lacking of recent documentation of ants. And ants are also declining because forest cover is declining, the land use pattern is changing, and we have no baseline information about the present status of ants. So our team started working on ants since 2018, and we recorded more than 50 species of ants present in 2 reserve forests and the campus of Guwahati University. Now I'm going to show the collections of the ants present in the Guwahati University campus, why the Guwahati University campus? It is a meeting place of different habitats. Like it has the natural hilly areas, natural wetlands, grassland areas, forest covers, as well as the man-made different wetlands, well-maintained botanical gardens, different concrete structures, and also nearby there is flowing the mighty river Brahmaputra, and there is a Ramsar site, that is Dipperville Ramsar site. So this particular area of 483.7 acre land is meeting site of different habitat, and here we got around 41 species of ants, which is far higher than the ant species that we recorded in the reserve forest. So these are the different sites where the ants are observed and collected in the grassland. It is present even in some wooden structures on the plants. We often see in foraging on the flowers. And we know that it is always a topic of debate whether ants actually play a role in pollination or act as a deterrent of the pollinators. So we recorded 13 species of ants which are foraging on the flowers, and also we observed different ant species that infested different wooden structures that is present in that particular habitat. And we also recorded the ants which have the co-evolutionary adaptation with the habitaran bugs. So there are 41 species. These are the under subfamily Dolichoderani. We collected and identified 4 species like Technomyrmex albates, Tapinoma melanocephala, 2 species of Dolichoderas. Then in the subfamily Elliptini, we recorded and collected one species of Anictus silonicus, which is a blind ant. We recorded 15 species of Formicini subfamilies. One species is very important, that is Zesomyrmex speculatus. Only one species has been reported from India, and that species is present in the campus of Gauhati University in the part of Assam. The first one is the Zesomyrmex speculatus, that is only species present from India. Then another is the invasive species also present. It is Anoplolis lepis gracilis. Paratrachina longicornis, which is the crazy ant, that is also present. Ocophylla sparatina, that we know that this is the weaver ant, and this species, the Asian weaver ant, also present in that particular locality. There are 5 species of Camponotus we have recorded: Camponotus albosparpus, Camponotus angusticolis, Camponotus comprisus, the carpenter ant, Camponotus mitis, and Camponotus spurius. There are 6 species of Polyrechis. This is the bicolor ant, that is Polyrechis bicolor, Polyrechis dives, Polyrechis elaudata, Polyrechis lactivenis, Polyrechis lavissima, and Polyrechis tibialis. These are from Formicini subfamilies. Now coming to the Myrmicini subfamilies, there are 11 species present, and there are 4 species under Chrematogaster genus. First one is Chrematogaster rogenhoperi, second one is Chrematogaster aberrans, Chrematogaster flava, Chrematogaster subnuta, and there are 2 species of Monomorium. One is Monomorium floricola, very familiar insect. Another one is Monomorium pharaonis, very small ant. Then Myrmicini subfamilies, we recorded another invasive species, Tetramorium bicarinata. And another important species present in that locality is Dilobucondyla gastroreticulata. This is the first report from our part. And from India, there are 2 species of Dilobucondyla. One species is present in our locality. Then Catalogus also present. We present here the species Catalogus temproveni, another bicolor, and Meranoplus bicolor under subfamily Myrmicini. This is Garipura diversal, another very common ant. Then under the subfamily Ponerini, we have collected 7 species. One is Platyteria parallela, another one is Botroponera tesserenda, 2 species of Leptozenis. One is Leptozenis birimana, another one is Leptozenis kiteli. And there's another 2 important predator ants, Dicama rubosum and Dicama scalpetrum. These are the predatory ants. And this one is also one species reported from India, that is Odontoponera. And we have also submitted the barcode of this particular species to NCBI, Odontoponera denticulata. So under the subfamily Pseudomyrmicini, there are 3 species. And these 3 species, mainly the Tetraponera elaborans, Tetraponera nigra, and Tetraponera rubonigra. Now, ants are the insects which are not often preferred by the predators because of their chemicals, the morphological adaptations, and therefore many species, other Archaeopteran species, they mimic ants. And we have studied the mimicomorphy that is present in this Archaeopteran species, especially in case of spiders. We recorded and collected 10 such mimicomorphic spiders from our locality. And mostly this spiders and nazis Myrmeracne that often mimic with this particular ant, that is Tetraponera rubonigra, Myrmeracne spider. So organized one exhibition and also compiled these things into a book, and I recently published it. it is Ant Fauna of Assam, case study of Guwahati University campus. And I want to acknowledge my mentors, Professor Arkabola, Professor Dipsika Bora present here, my mentor, and Professor S.R. Hassan and Hemendra Bharti from Punjab University, India. And also I want to acknowledge Professor J.C. Colliton, the head of the department of our university and Department of Biotechnology, Government of India, for giving the initial support to carry out the work. Thank you. Yes, sir.
Did I hear you say you were doing some barcoding in DNA?
Yes, all of them were that curated, that is published in Zoologist or Elsevier. Yes, madam.
Plans to upload your specimen records and species records to an online database?
You can go for that, but we have not yet got any, somebody approves like that, then we can discuss on that.
Well, thank you very much.
Thank you so much.
So due to the cancellation, we will actually have time for a very short coffee break, which is great because in the schedule previously we didn't. So we have 9 minutes before we're going to start the next talk. So if you'd like to mingle, grab yourself a coffee, and then we will start back at 8:55 local time. So is it for Jeff? And this is presented by Delape and Mello.
Buenos días, mi nombre es Pablo de Leopé, soy el jefe de la división Entomología del Museo de La Plata. El Museo de La Plata fue creado en 1884, 2 años después de la fundación de la ciudad de La Plata, nueva capital de la provincia de Buenos Aires, y abrió sus puertas al público 4 años después. Es el primer museo de Sudamérica en construirse con los propósitos específicos de museo, con un diseño neoclásico combinado con ornamentaciones inspiradas en las culturas meso y sudamericana. Desde 1906 pertenece a la Universidad Nacional de La Plata y durante muchos años funcionó como centro científico y escuela de ciencias naturales, hasta que se creó la Facultad de Ciencias Naturales en 1949. Actualmente es un museo con exhibiciones de historia natural y un centro científico con 5 áreas de investigación: paleontología, geología, botánica, antropología y zoología. Las colecciones fundacionales del museo provinieron del Museo Arqueológico y Antropológico de Buenos Aires, creado por Francisco P. Moreno en 1877, el cual albergaba materiales colectados durante su infancia y preservados por su familia. Francisco Moreno fue el primer director del Museo de La Plata y quien contrató a especialistas europeos para organizar las diferentes colecciones y a naturalistas viajeros para realizar expediciones a diferentes regiones de la Argentina con el fin de recolectar materiales de estudio y de exhibición. Algunos de ellos son reconocidos estudiosos de sus disciplinas, como Carlos Ameguino, de mamíferos fósiles de Patagonia y La Pampa, Adolfo Metzfessel, pintor suizo, Carlos Bruch, naturalista alemán, Carlos Spegazzini, botánico italiano, Clemente Onelli, paleontólogo, o Fernando Laille, zoólogo francés, entre otros. La colección entomológica tuvo su origen a partir de la donación de la colección privada de Carlos Bruch en 1896, y desde ese momento se fue enriqueciendo por el trabajo de naturalistas contratados por el museo, por donaciones y por compra de colecciones privadas. La colección de Carlos Bruch incluye principalmente coleópteros y formícidos, y entre las colecciones más importantes podemos mencionar la donación de la colección de lepidópteros de Paraguay de Pedro Jorgensen, la adquisición de la colección de Juan Tremoleras de Uruguay, constituida principalmente por coleópteros, la colección personal de hemípteros de Carlos Berga y la colección de mariposas de Alberto Breyer. También podemos mencionar la colección de Juan Bosch, constituida principalmente por insectos perjudiciales para la agricultura, o la colección de coleópteros de Manuel Viana. Actualmente estimamos que la colección entomológica del museo incluye 2,5 millones de especímenes y más de 10.000 especímenes tipo, los cuales están en su mayoría catalogados en los 53 catálogos publicados a la fecha. Estos catálogos se encuentran digitalizados y disponibles en el repositorio digital de la Facultad de Ciencias Naturales y Museo, Naturalis. La colección entomológica depende de la división Entomología, que es una de las 15 divisiones científicas del Museo de La Plata. En la colección trabajan 2 técnicos a tiempo completo y 3 encargados de colección con dedicación parcial, que se ocupan de tareas de mantenimiento, curaduría, digitalización y gestión de la colección. Todo este personal está a cargo del jefe de división, quien organiza las tareas y delega parte de ellas en los jefes de sección, que son investigadores activos de la división de los taxones bajo su cuidado. Las 7 secciones científicas existentes abarcan gran parte del material de la colección: Coleóptera, Díptera, Hemiptera heteróptera, Hemiptera auquenorrinca, Hymenoptera serie parasítica, Hymenoptera serie aculeados y Ortopteróides. Los grupos taxonómicos que quedan por fuera, como Lepidoptera, Tisanoptera, Tiráptera, etc., se encuentran bajo la directa responsabilidad de gestión del jefe de división. La colección entomológica está localizada en el primer piso del museo, en 2 sectores, separados de acuerdo con los estándares de conservación y medidas de seguridad establecidas. La colección seca tiene control de temperatura y humedad, 20 grados centígrados y humedad del 50%, y la colección húmeda está en otra habitación, que también es utilizada por curadores, técnicos y también por visitantes si es necesario. Nuestra colección tiene una importante representatividad de la fauna entomológica argentina y de países limítrofes, aunque en algunos grupos en particular hay variada representatividad de otras partes del mundo. Actualmente las colecciones crecen gracias al aporte de investigadores que trabajan en el museo y a partir de los viajes de campaña que se llevan a cabo durante el desarrollo de proyectos de investigación. Previo al ingreso de nuevo material a la colección, y particularmente para el montado de alfileres entomológicos, se lo somete a una semana de cuarentena en freezer. Este mismo procedimiento se aplica para las devoluciones del material que fue prestado. Además, se registra en planillas de depósito todos los datos de los ejemplares para luego ser importados en la base de datos de la colección. La mayoría de los especímenes de la colección seca están montados en alfileres entomológicos o en preparados microscópicos y una menor cantidad en showers entomológicos o en camas de algodón entomológicas. La colección de insectos pinchados se encuentra mayormente en muebles metálicos y cajas de Revit y en algunos otros muebles más modernos con cajones y unit trays. En el futuro se planea expandir en esta modalidad de guardado y eventualmente reubicar lo anterior. Las colecciones con mayor cantidad de especímenes y diversidad taxonómica son las de Coleóptera, Lepidóptera e Hymenóptera, aunque también las colecciones de Ortóptera y Hemíptera Heteróptera son importantes, dado que hay investigadores especialistas en estos grupos que han enriquecido estas colecciones en los últimos años. Asociada a la colección, mantenemos una biblioteca con los trabajos que mencionan el material depositado en la colección, la cual está siendo reorganizada y digitalizada. Esta biblioteca se está incorporando en una base de datos bibliográficos utilizando Zotero y está organizada en diferentes colecciones, catálogos, grupos taxonómicos, materiales tipo, etc. Esto nos permite rastrear posibles materiales faltantes que debieran estar ingresados en la colección y gestionar su reclamo y devolución o ingreso en la colección. Además, desde hace varios años, se lleva adelante la digitalización de las colecciones del museo como parte de un proyecto institucional. En este sentido, se han ejecutado diferentes proyectos y particularmente en la división de entomología se trabajó en la confección de planillas Excel con los datos de los especímenes tipo, en el estándar Darwin Core y su posterior puesta a disposición en GBIF. Sin embargo, debido a su magnitud, esto representa solo una pequeña proporción del material de la colección. Actualmente, los encargados de colección están trabajando en el ingreso de datos de especímenes de colección general con el software Specify y de acuerdo a la clasificación de Catalog of Life. Actualmente contamos con registro de alrededor de 40.000 especímenes de colección general y se está trabajando activamente en la adecuación de planillas Excel provenientes de proyectos previos para su importación a Specify, lo que constituye unos 20.000 registros extra, además de los 10.000 especímenes tipos que ya actualmente están disponibles en GBIF. Además de su uso como herramienta de gestión interna de la colección, parte de esta información será disponible online en un futuro cercano a través de la página web del museo. Muchas gracias.
I'm just going to see if Pablo is online. Yeah, so he is online. So if anyone has a question, we have time for one or two. And it can be in English or Spanish because we have Jen to translate. Well, if there is no questions in the room, is there any online that you can see, Ash? Well, thank you very, very much for sharing that fantastic collection. And next up, we have the Museo de Zoológica, Zonas, an emergent museum in southern Ecuador.
Good morning. My name is Aura, and I am the curator at the Entomology Division of the Museo de Zoología Lonas. A little bit of background. This museum is located in southern Ecuador at the Universidad Nacional de Loja. It was established in 2007. At that time, the main focus was on vertebrate fauna, and its invertebrate collection started at the end of 2019, with a pause due to the pandemic. So it is a fairly new invertebrate collection. However, due to the museum projects on insect ecology and conservation, the collection has grown to have over 17,000 pin insects. It also has a wet collection of near 40 lots of microinvertebrates from environmental impact studies in the region. Part of the collection data has been placed in the GBIF Ecuador node with a projection to make available all the museum database in the following years. The collection also holds 66 type specimens. Our first types were deposited in April of this year. For that, we organized a big event to receive our first types, which were the holotype and allotype of the scarab beetle Chrysina mercedes, which was described by Manuel Barria from Panama. There is Manuel in the picture. Soon after, other types follow as the holotypes of Hymenoptera and Mollusca. The museum also holds a collection of over 100 isolated bacteria strains associated with dung beetles. This collection is projected to expand to other taxa with the mission to find ecological relationships and ecosystem services to restore degraded habitats. I would like to show you a video of the museum facilities. We are at Universidad Nacional de Loja. This is the building of the environmental engineer career where the LONAS Museum is located. So let's go see it. We are located on the first floor of the building. These are our offices. There is the lab, and there is the collection room. Here we have part of an educational collection that we used to teach biology classes for students at the university, but also for students at elementary schools and high schools that visit us. At the museum, we mainly hold Southern Ecuador material, although we also have donations like this butterfly collection from Esmeraldas in Northern Ecuador. We hold a scarab collection. These are some dynastines which were included in the book, The Dynastines of Ecuador. There is our logo. And here is the book. Now, let's head to the collection room. So this is the collection room. This museum holds collections of research done on plant insect interaction, insect conservation. We have also done some research on light pollution, scarab distribution, and we have created also or developed local guides. This is part of the scarab collection. Here we see some dynastines and megasoma. There are some golofa, of course. They're very common here in Loja City. And this is part of a larger collection of dung beetles from the Ecology in Natural Forest and Livestock Landscape Studies we have done here at the museum, developing many theses with students. Last month, the LONAS Museum received a donation of a collection held in the agronomy department at the same university that we see here. This is in a different building. Here we see a collection of ichneumonids and other hymenopterans. Outside here also other ichneumonids from long-term studies on bioindicators. Over here, there's some other interesting specimens we are receiving that we will be curating. We also have to database this material and it will be included in our collection. In this area, we also have some historic collections from some of the first entomologists at the university. As we are growing, we are transitioning, and this is the space we are moving into fairly soon. I believe the museum has filled a gap cataloging and understanding biodiversity in the southern region of Ecuador. And it has provided a great scenario where students acquire new understanding of the natural world and start to appreciate the importance of conservation and the need for sustainable management of biodiversity to bring solutions to the region. I would like to thank all the people who are involved at the Lunas Museum Achievements that due to the time, I cannot say all the names, but my gratitude goes to our museum researchers, to our collaborators, also to other national museums we have built a network with, and also a very special thanks to our students. And thank you for listening.
And I believe that Aura is also online joining us. Yes, so if we have any questions in the room or online. Yes, sorry.
How is the beetle data and the bacterial data associated?
So the question in the room is, how is the beetle data and the bacterial data associated in the collection?
Hello everybody, good morning. We have studies that we have been developing lately. So for the collection, we decided to keep these bacteria associated with it by storing them in a -80 degrees freezer, but also part of the museum will have to legally develop a separium in order to keep these two collections together. We think it's important because we don't want to lose or separate this information and lose all of these data that we have now. So that's the idea, to keep it together as part of the Lunas Museum. The bacteria associated with bugs and also the bug collection.
That is extremely fascinating. Any other questions in the room or online? Well, thank you so much for joining us virtually and showing off your beautiful new collection there.
Thank you.
Next, we have a tour from Colombia. This is the Lepidoptera and Euglossini de la Colección de Museo de Histórica Natural de la Universidad del Cauca.
Buen día para todos, mi nombre es María Cristina Gallego Ropero, soy profesora del Departamento de Biología en la Universidad del Cauca, investigadora y curadora de la colección de entomología adscrita al Museo de Historia Natural de la Universidad del Cauca. Inicialmente me gustaría contarles a ustedes que el Museo de Historia Natural, adscrito a la Universidad del Cauca, fue fundado en 1936 y alberga colecciones de la fauna colombiana principalmente. Este museo, en el año 1983, sufrió una gran pérdida, dado que para esa época ocurrió en el país, y particularmente con epicentro en la ciudad de Popayán, un terremoto que además de destruir gran parte de la ciudad y generar grandes pérdidas humanas y materiales, también afectó fuertemente el museo, que en aquella época se encontraba en otra sede diferente a la que actualmente se encuentra. Para ese entonces, la universidad decide trasladar lo que se recupera de ese museo para las instalaciones, las antiguas instalaciones de lo que eran las residencias universitarias. Entonces, desde 1985 más o menos, 84-85, la nueva sede del museo es la que actualmente tenemos. Importante también contarles que en el caso de la colección de entomología, fue una colección que se dio inicio más o menos en la época de 1987, de acuerdo con lo que cuentan los registros, y fue iniciada por el profesor, que en paz descanse, Álvaro José Negret, quien en 1996 falleció. Desafortunadamente, esta colección pequeña, pero interesante para la región, estuvo durante mucho tiempo sin un liderazgo, sin un proceso de curaduría, sin alguien a la cabeza. Pero bueno, en la última década, afortunadamente, hemos, bajo mi liderazgo, coordinado el proceso de curaduría de esta colección que hemos ido alimentando cada vez más con la formulación de proyectos de investigación, en la cual han participado estudiantes de pregrado y de posgrado, también la comunidad de la zona de influencia donde se ejecutan los proyectos, y hemos ido fortaleciendo esta colección cada vez más. Es importante notar que uno de los proyectos que más peso le ha dado, digamos, más fortaleza le dio a esta colección, es un proyecto. En el 2016 se aplicó a la convocatoria, en el 2017 ganamos, sobre una expedición llamada Colombia Bio, y en este caso Colombia Cauca. Este proyecto nos permitió recorrer gran parte del territorio del departamento del Cauca, particularmente en la zona que consideramos muy importante y estratégica para la biodiversidad, como es la conocida bota caucana. En ese orden de ideas, ese proyecto también nos permitió, además de ir a hacer trabajo de campo muy interesante y recorrer estos territorios que han sido muy afectados por el problema de orden público que hemos vivido en este país, también nos permitió comprar particularmente un compactador para poder adecuar la colección con las normas internacionales. El compactador, algunas pocas cajas entomológicas, materiales pues de consumo básico, cajas guarda, alfileres entomológicos, en fin, lo que ya todos conocemos, y eso nos dio un empujón grande para la colección. Bueno, es importante también aclarar que esta colección que se ha alimentado en los últimos años, como les mencionaba, tenía particularmente en el grupo de mariposas mucho material guardado, ¿no es cierto?, en sobres de papel mantequilla engavetados. Material que en los últimos 2 años particularmente, con todo el apoyo del trabajo, digamos, muy juicioso y minucioso de los estudiantes que pertenecen al semillero y también voluntarios que llegan al museo, entrenándolos a ellos para el proceso de montaje y de curaduría, digamos, hemos logrado sacar adelante esa cantidad de material que incluso en ocasiones tiene pocos datos de la localidad de colecta. Pero bueno, es un proceso que se ha ido adelantando y que finalmente estamos casi terminando. Desde el punto de vista de la colección de las abejas, es una colección que realmente es mucho más incipiente, bueno, era mucho más incipiente hasta más o menos hace unos 7, 8 años, cuando empezaron precisamente, como les estaba comentando, a trabajar estos proyectos de investigación. Esa colección en este momento se ha incrementado bastante, ostensiblemente. Hemos además de participar en una expedición de Guainía, donde también se incrementó los datos para mariposas, para otros grupos de insectos como los coprófagos, y también en este caso particular también de las abejas de la tribu de Glucina. Bueno, entonces es importante también que entendamos que esta colección es una colección muy, muy concentrada hacia la parte suroccidente del país, es decir, el departamento del Cauca y sus límites, particularmente hacia el sur, con Nariño y digamos suroriente, con la región del Caquetá y la región del Putumayo también. Una zona muy interesante porque es una zona de transición andino-amazónica, entonces estamos en los Andes, tenemos del lado oriental la zona amazónica, pasamos por los Andes y luego pasamos a la zona pacífica. Entonces es muy interesante estratégicamente por la diversidad de ecosistemas en los cuales nos encontramos. También puedo mencionarles que hemos contado con todo el apoyo de los estudiantes, pero además de eso hemos contado con la suerte de tener visitantes, hemos digamos hecho interacción con otros investigadores a nivel nacional e internacional, y particularmente con taxónomos a nivel nacional, con el profesor Alfonso Villalobos Moreno, y a nivel internacional con el profesor Ismael Hinojosa de la UNAM de México, con la profesora Fabricia Freitas de Olivera de la Universidad de Bahía, Federal de Bahía, y con el profesor André Nemesio de la Universidad Federal de Uberlândia. Profesores de Brasil que son taxónomos junto con el profesor Hinojosa de México y con el profesor Alfonso, y también en colaboración con el profesor Julián de la Universidad de Caldas, con Lecrón, con Miguel Constantino, digamos taxónomos reconocidos a nivel nacional que nos han apoyado en ese proceso de confirmar las identificaciones de los especímenes de las mariposas hasta especie. Y en el caso de la profesora Fabricia, el profesor Ismael, y en el caso de Nemesio, la confirmación de todos los especímenes que hemos colectado, y estamos aún en proceso de curaduría, ¿no es cierto?, montaje y curaduría completamente y catalogación para ingreso a la colección por parte de la taxonomía por parte de ellos. Bueno, entonces también es interesante contarles que nosotros, digamos, a grosso modo, también se han adelantado algunas publicaciones, pues finalmente la meta también de poder trabajar en las colecciones biológicas en los museos es que se hace muy importante compartir ese conocimiento que de entrada cuando se ejecutan los proyectos y se formulan, particularmente nosotros desde el grupo de investigación y semillero, trabajamos directamente con la comunidad, las comunidades, digamos, nos apoyan en el proceso de como investigadores de campo y aprenden también, y nosotros aprendemos el conocimiento y ellos comparten con nosotros nuestros conocimientos científicos. Entonces, aquí también les puedo mostrar algunas de las publicaciones que hemos hecho. Ya hemos trabajado pues en una aproximación de las mariposas del departamento del Guainía, pero también estamos organizando muy bien, como se pueden ustedes observar en las fotos. Hemos hecho todo ese proceso de curaduría y como pueden ver aquí, estamos viendo la colección de los papilionios ya organizada, la colección de todo lo que es ninfálide con los diferentes grupos internamente, porque es una de las familias más grandes que encontramos dentro de los lepidópteros. Pero también estamos trabajando con la familia Perry. Aquí, en todos estos procesos, estamos involucrando trabajos de grado, de estudiantes de pregrado y estudiantes de maestría. Entonces, aquí eso es parte de las publicaciones que hemos hecho hasta el momento. Tenemos unas también sometidas en este momento que aún no hemos tenido, pues estamos en el proceso de revisión. Bueno, entonces eso es todo por hoy. Muchísimas gracias por estar aquí. Los invitamos a que conozcan nuestra colección de entomología del Museo de Historia Natural. El museo, además de eso, cuenta con una colección, digamos, de exhibición en los diferentes grupos y también en el caso de los insectos. Y los invitamos a apoyar este proceso porque necesitamos conseguir recursos para poder seguir trabajando y sacando adelante esa colección que realmente es muy importante, es patrimonio biológico, ¿no es cierto?, de la humanidad, no solamente para Colombia y el departamento del Cauca. Muchas gracias.
And I think Maria is also online. Perhaps.
It's under a different name.
María Cristina, ¿estás?
Well, unfortunately, Maria is unable to join us, but it was a fantastic insight into the Colombian collection there. So next up, we have a presentation on the catalog of type material deposited in the entomological collection at de Mato Grosso in Brazil.
Olá a todos, então, eu me chamo Emanuel Rodrigues Vivas Gama, eu gostaria de primeiramente agradecer pela oportunidade de estar aqui apresentando nesse evento, gostaria de agradecer em nome também dos meus colegas, e eu vou estar apresentando um pouco aqui sobre esse trabalho que nós estamos desenvolvendo, eu juntamente com Jorge Armando Arias Buritica, Diego Felipe Rodrigues e com o professor Fernando Zaguri Vaz de Mello, que é esse catálogo, né, esse trabalho intitulado Catalog of Type Material Deposited in the Coleção Entomológica de Mato Grosso, Eurides Furtado, CMT, Cuiabá, Mato Grosso, Brasil. O número de coleções biológicas vem aumentando consideravelmente no Brasil nos últimos anos. E segundo esse banco de dados que nós temos, né, de coleções registradas, o CIBBR, nós temos atualmente 283 coleções que tratam de fauna no Brasil, né, sendo 39 dessas focadas na entomofauna brasileira. O setor de entomologia da coleção zoológica de Mato Grosso, da Universidade Federal de Mato Grosso, ele é uma dessas coleções. Essa coleção foi fundada em 1987 e essa coleção começou como uma coleção regional. Essa coleção está localizada, então, no estado de Mato Grosso, na cidade de Cuiabá, dentro do campus da Universidade Federal de Mato Grosso. Essa linha em vermelho, então, na imagem mais abaixo à direita, corresponde à área do campus, e a linha amarela corresponde ao prédio onde a coleção está atualmente. Até 2008, essa coleção era uma coleção de cunho regional, né? Trabalhava apenas com espécies da região aqui, e apresentava 12 gavetas. E essa coleção, ela está numa região muito interessante, porque ela é uma região onde a gente consegue encontrar espécimes de 3 biomas distintos, que são a Amazônia, Cerrado e o Pantanal. Entretanto, de 2008 para cá, a coleção ela tem sido curada pelo professor Fernando Zaguri Vaz de Mello, que é um especialista em escarabeide, né? Então, a partir do momento que ele assume essa coleção, que é em 2009, ele não apenas incorpora sua coleção pessoal de besouros, que já era uma coleção grande, como também ele muda o objetivo central da coleção, que deixou de ser uma coleção regional para virar uma coleção de referência em coleóptera. Ele também fundou o Laboratório de Escarabeidologia. Em 2010, então, a coleção já tinha aumentado consideravelmente, o número de gavetas já tinha aumentado muito, e o número de exemplares, então, depositados também já havia aumentado bastante. Atualmente, em 2023, a coleção conta com 2 armários fixos e 3 armários móveis para armazenamento de gavetas, que correspondem ao total, então, de 2.640 gavetas, mas nós temos 300 gavetas que ainda não podem ser adicionadas a esses armários, né, por falta de espaço. Então, nós temos 300 gavetas que estão prontas, mas que não foram adicionadas à coleção ainda. O nome da coleção mudou recentemente, então, para quem já conhecia, que passou de ser o setor de entomologia da Coleção Entomológica de Mato Grosso, a coleção agora é intitulada Coleção Entomológica de Mato Grosso, Eurides Furtado. A mudança do nome da coleção foi em homenagem a um pesquisador, a um lepidopterólogo, grande amigo do professor Fernando, um grande pesquisador. Esse pesquisador era, então, o professor Eurides Furtado. Ele veio a falecer em 2022, e após o seu falecimento, toda a sua coleção de lepidópteras, né, foi incorporada à coleção entomológica de Mato Grosso, Eurides Furtado. Hoje, então, a coleção conta com mais de 700 mil espécimes preparados, mais de 140 famílias de coleópteros de todo o mundo estão presentes nessa coleção, e mais de 24 mil espécies de coleópteros brasileiros estão presentes nessa coleção. Existem aproximadamente 7.500 espécies de escarabeoide na coleção, né? E mais de 6,5 milhões de espécimes ainda aguardam serem preparados. O objetivo do presente trabalho, então, é criar um catálogo amplo com todo o material tipo que nós temos depositado na coleção. A gente fez esse trabalho de 2 formas. Primeiramente, a gente buscou na literatura pelos acrônimos da coleção entomológica de Mato Grosso e pelos acrônimos da coleção pessoal do professor Fernando. Então, esses acrônimos foram CNT, o FMT, que é o acrônimo para a Universidade Federal do Mato Grosso, FVZC ou FZVM. E nós também buscamos ativamente por tipos, né, nas gavetas da coleção, porque alguns tipos são mais antigos, né, e podem não apresentar esses acrônimos. E o que nós fizemos? Ao encontrar esses tipos, nós transcrevemos os dados nas etiquetas desses espécimes e desses líderes, né? Escrevemos sobre como a etiqueta ela se parece fisicamente e adicionamos uma etiqueta com o número tombo e com o QR Code, e esse material foi incorporado, então, ao banco de dados da coleção. Nossos resultados preliminares, então. Material tipo por ordem. São 6.096 espécimes, tipos de coleóptera, 4 de díptera, 1 de himenóptera e 1 de neuróptera, que somam 6.102 exemplares tipos, então, né? Destes, 185 são tipos primários, 184 são de coleóptera, 1 de díptera, 2 lectótipos de coleóptera, 3 neótipos de coleóptera, 3 paralectótipos de coleóptera, 5.911 parátipos de coleóptera, 2 parátipos são sinônimos, então, né? E um síntipo. Para díptera, então, nós temos um holótipo de 3 e 3 parátipos, e Menoptera e Neuroptera, um com cada parátipo, cada um com um parátipo. Então, isso soma um total de 5.897 parátipos. E como a gente falou, deu para ver anteriormente, né, a maior parte do material tipo que nós temos nessa coleção é de Coleóptera. E esse material, essa abrangência, esse número de tipos se deve principalmente a 3 famílias de Coleópteras distintas. São elas Scarabaetes, um total de 4.534 tipos, sendo 123 tipos primários, né? Um lectótipo, 2 paralectótipos e 4.408 parátipos. Em seguida, nós temos Melolontides, com 602 exemplares tipos, sendo 25 holótipos, um paralectótipo e 574 parátipos, e dois sinônimos, né? Parátipos e sinônimos. E Geotrúpide, com um total de 429 exemplares tipos, sendo 9 holótipos, um lectótipo, um neótipo e 416 parátipos. Esses são resultados preliminares, do que a gente ainda está buscando tipos na coleção, e a gente pretende, então, trabalhar também com o material de Lepidoptera, que foi posteriormente introduzido graças à coleção do professor Eurides. Como a gente está falando de futuro, né, então a gente tem que aproveitar para falar que a coleção ela vai ser movida para um prédio especializado, né, dedicados às coleções zoológicas da UFMT, e isso ocorrerá no futuro próximo. Bom, muito obrigado pelo tempo de todos, né? Eu agradeço meu nome, né, em nome dos meus colegas, como eu falei anteriormente. E aqui está o e-mail do coordenador do trabalho, né, o Jorge Arias. Para quem quiser conversar, aqui está o e-mail dele à disposição de todo mundo. Então, muito obrigado pelo tempo. E é isso.
And is Jorge online here?
Yes.
Is there any questions in the room or online? I was extremely impressed with was it 6.5 million unprepared scarab beetles? What is the plans for them? Is there a bunch of students? Is there any effort to get them prepared?
Respondo en español. Yo soy colombiano, estoy encargado de este proyecto. Bueno, el futuro es que todo este material esté montado, obviamente depende de estudiantes, de proyectos que se están desenvolviendo, pero básicamente en este momento estamos con un proyecto grande de Amazonía que está incorporándose todo el material directamente y los otros tienen que dar una espera. Estamos trabajando en eso, pero pues es mucho material que hay por montar y que estamos trabajando poco a poco.
So he said he's from Colombia and he's now in charge of processing all of this material. So some of it is being processed by students, but they are focusing right now in a big project that they have in the Amazon region. So the material that is not from that region is going to be processed later at some point, but the hope is that everything gets mounted, labeled, and processed all the way. Any other questions?
Thank you so much for joining us. So unfortunately, the next tour from Argentina has also been withdrawn, but we did miss a question online earlier that I think was for the Lepidoptera collection for the Natural History Museum. Ash, do you want to, Jeff, if you're in the room still? He just went out. Sorry.
I think we'll just keep going and have a longer break between parts, yeah.
But if no, okay. Well, we might just move on so then we can have a longer break in between the two sessions. So So the following tour is from Honduras at the insect collection at Zamorano.
Hello, my name is Jesus Orozco. I'm the curator of the entomology collection at Zamorano, Honduras, and today I'm going to be presenting about our institution and our collection. Just to put you in perspective, Zamorano is an institution located in Honduras, also known as the Pan American School of Agriculture. There's 4 departments, 4 programs are offered to the students, all in the science of agronomy. It's a vast institution. It covers a lot of land, about 10,000 acres. And since its beginnings in 1945, it has graduated close to 9,000 students from 30 countries. There's a lot of nationalities in Zamorano. Students come from all over Latin America and sometimes from other countries like Italy, Japan, Korea, and we had students from different places. There's something particular about Zamorano. Zamorano is a boarding school. It's a co-ed boarding school. Students live on campus and they work half of the time in the fields, in the labs, taking care of the crops, and the other time they're taking courses. So it's an intense program of 4 years. And there are 21 university learning and production units that where the students get the opportunity to participate. And that's where the insect collection is located. Our insect collection started, and some of you probably know this guy, and probably he's attending the meeting today. The collection started at the beginning of the 80s from material collected by the students from the entomology class. Keith Andrews was the person that started the collection from this material and from other material from boxes from the students and from other projects related to IPM that were occurring at the time. In 1987, Ronald Cave, the person in the picture, came to Zamorano, got money to put the collection in a new building and organize the collection by taxonomy. Previously, the collection was only organized depending on the insects that were present or not in a crop. He put a lot of effort into collecting insects all around Honduras, and more than 300 localities were sampled. And the database was started in 1998, or Subtra database. 326 types are part of the collection now. And there has been other curators that came before. Ronald left Zamorano, but the biggest contributions were done by him. Currently, the collection has got 38 cabinets, and to no surprise that most of the material belongs to the Coleoptera. Ron was a specialist in Coleoptera and Hymenoptera, and that's the reason that there are so many of them in the collection. Scarabeids are well sampled in Honduras. There's a good collection of Cerambycids also, and there's also good material of Microhymenoptera. And related to Lepidoptera, there we have a very good collection of Sphingids also. Not all of our cabinets are these new type of Canadian cabinets. Actually, most of the collection is in these very well used and very practical older brownish cabinets. And all of the material in the collection is organized in Cornell drawers with tray boxes. Said that, we have working cabinets also where we don't have Cornell drawers, and we have different types of boxes, and tray boxes are sometimes difficult to get for us down here. So, but what is all the reference material? It is in Cornell drawers with tray boxes. We're reaching the 250,000 specimens in the collection. slowly we're absorbing Jean-Michel Maes' collection of insects from Nicaragua. We've been getting that material slowly for the last 8 years or 10 years. And most of what we have in Zamorano belongs to the Coleoptera, followed by Hymenoptera and Hemiptera. And all those small orders at the end of the graph has been we set up to get more of that material so they are better represented in our collection. When it comes to identification, most of the material is identified to family level, as you can see in the upper left graph. Dermaptera is 100%, Coleoptera very close to 100%, the mantids also very close to 100%, and it goes down. Lepidoptera, we're close to 70%, and Diptera also around that number, and Neuroptera is the order where we don't have identified to family level maybe half of the material we have. When it comes to identify to genus level, which is the upper right graph, Dermaptera is the order where we have most of the material, at least to genus, followed by Coleoptera, Odonata, and as you can see, Lepidoptera is also well identified. When it comes to species level, Dermaptera, there's a low diversity, is the order best represented with identifications to the species level with about 75%. But if you look at the Coleoptera, about 55% of the material is identified to the species level, which means there's a lot of work to be done. Orthoptera, very, very poorly identified to the species level. Also Diptera, very important orders in the region, and Hymenoptera also very, very poorly identified. We also have a wet collection with around 20,000 specimens, which need a lot of work in the curation. And there's also a slide collection, about 103 slide boxes with close to 5,000 specimens. And this is something we've been putting effort in the last couple of years. So all the projects and the stuff we do here in Zamorano is done by undergraduate students. And since 2015, which marks the time of my arrival to Zamorano, we had students doing projects in entomology, 62 students in total from 10 countries. And some of them continue to do their work in the United States as master's students or PhD students, and some already have graduated and are working at important companies. And this is just an example of some of the students that have been doing the projects. And we've done projects that go from through bugs to Hymenoptera to bees. There's projects that we keep doing and started doing in 2018, like the flies of forensic importance. They're very relevant for the region, and we want to learn more about the Diptera. And we've done projects also on dragonflies. Dung beetles is also persistent. The research we've been doing on that. And in 2019, we started working on the black soldier fly, which is Hermetia illucens. And that doesn't relate to taxonomy or collections, but it's something that got a lot of the students involved in entomology. So we consider that to be a big part of importance for our lab, and we continue doing it. Some of these projects have resulted already in publications. For example, the work we did on tabanids. I think we have 11 new records for the country, and we're working on big manuscripts on tephritids and Eulididae. A lot of new records for the country, and we're working on manuscripts on thrips. So there's a lot of stuff that is happening, exciting stuff. And this year we started with this colony of Sophobas morio also. We did some projects on Q-lizards and white flies. What we want to do next year was we're going to start working on termites, on ants, and on aphids. All three groups poorly represented in the collection, and groups that need to be done, and we need to know more about them and the students are the ones in charge of this. And this is the team. This is the people that works in the collection right now. Catherine Veroy is our collections manager. She's a microbiologist by training, by entomologist by heart, and she's been with us 5 years now. And Rosa Ortega has been in Zamorano for 37 years. She's the heart of the collection, and of course, you got me on the right. I've been here for 8 years. And that's 8 minutes. And yeah, well, with that, I want to just thank you very much for the invitation, and that's my contact information, and I would love to hear from you if you have any questions.
No, do we have his online? Yes, we do. So does anyone have any questions in the room? I just saw.
I thought, oh, yeah. Yes. Oh, here's a question on chat.
There's a question online, and then there's also one in the room. So if we just read the one that is on. Is this collection integrated with the agricultural mission?
Oh, yes. Hello, good morning to everybody. So most of what we do is related to pests and biological controls in the university. And when it comes to scale insect identification, we're doing diversity. We don't know what species we have in Honduras, especially in Zamorano's campus. We have some crops that are being attacked by scale insects. So we're going to try to identify the stuff that is causing us damage.
We have a question in the room, yes?
I was just wondering if the non-shell collection from the Bravo is separate from the Honduran collection?
So the question is, the collection from Nicaragua kept separately from the Honduras collection?
When it comes, yes. But it was slowly integrating both collections into the main collection.
Great. And I also have a comment. It was fantastic to see so many undergraduates involved in this collection. How do you attract them into working with you?
Well, I teach the entomology course, so I try to be very enthusiastic, which is very easy with insects. And then they decide to do the project with me.
It seems that you're extremely successful at it. So thank you very much for your wonderful tour and for joining us online. So to wrap up the collections tours today, we have one from the University of Kentucky Insect Collection.
Oh, you startled me. Are you here to learn about insects? All right, let's go. My name is Julian Dupuis. I'm an assistant professor in the Department of Entomology at University of Kentucky and curator of the University of Kentucky Insect Collection. We're here in Lexington, Kentucky in the Dimmock Animal Pathology Building on UK's main campus, built in 1942 and still holding strong. I got a hunch about you. You a little bit of a history buff? You want to learn more about the collection's history? That's what I thought. Let's do it. The collection goes back to the 1880s and Harrison Garman, our first curator and the first chair of the Department of Entomology at University of Kentucky. And if I do say so myself, a pretty dapper-looking gentleman. Now, Harrison Garman's specimens were interesting in that they leave a bit to be desired. He labeled each individual specimen with a single numbered label, and that number relates back to his logbook, where he more or less had complete locality information written down. Garman collected extensively in those early years. By 1894, there were over 24,000 pin specimens and 40,000 specimens in alcohol in the collection. In 1905, the collection was moved from its original location to the University of Kentucky Agricultural Experiment Station, now called Scoville Hall on UK's main campus. The move was led by Mary Didlake, a departmental assistant and later interim chair. And she notes in her journals that the move was conducted on a snowy, blistery winter day, and they actually used farm sleds to move all of the specimens across campus to its new location. After Garmin, the collection has had 4 curators, including myself. And I'd be remiss if I didn't mention Dr. Charlie Covell, who curated the University of Louisville collection. The University of Louisville collection was integrated into our main collection in 2003 and has approximately 250,000 of our estimated 1 million specimens that we have in the current University of Kentucky insect collection. Speaking of numbers, you want to hear a little bit more about the holdings in the UK insect collection? That's the spirit. Let's go. We estimate that the UK insect collection has upwards of about 1 million pin specimens. Our alcohol holdings are definitely the least organized part of the collection, so we're a little more unsure of our numbers there. Now with pin specimens, specimens are stored in 3 different ways in the UK insect collection. In wooden Schmidt boxes, the original method of storage in the University of Kentucky insect collection, and actually in 2 sizes of standard insect drawers, both USNM and Cornell sized. Now, despite having consistent curators since the 1880s, there's been little curation activity in the UK insect collection in the last 20 or 30 years. One of the biggest challenges and efforts that we're currently undertaking is taking these 3 independent subunits, original wooden Schmidt boxes, USNM drawers, and Cornell drawers, each that have their own taxonomic organization, and integrating them into one taxonomically cohesive unit within the collection. That involves taking specimens from each of their individual sources, figuring out what size drawer they belong in, and then moving them to that location. Whew! Talk about a lot of work. Now, in addition to integrating these different collection subunits together, we've also started digitizing and databasing specimens within the UK insect collection. Now, who here is a fan of beetles? I figured as much. The collection manager here at the UKIC, Dr. Eric Chapman, is also a fan of beetles. Eric, break the 4th wall here. So obviously, we started our digitization efforts within the Coleoptera. Now, Kentucky's a bit of an interesting state in that it's kind of a black hole for specimen records. When you look at a lot of old genus level revisions, Kentucky specimens will be completely absent, like this example. Talking to curators from multiple collections around North America, they often anecdotally refer to the fact that, yeah, I don't really see many Kentucky specimens in our collection. And even looking at broad groups like all of the odonates and modern collection digitization efforts, you end up seeing maps looking like this. Now, for those geographically inclined folks, you might see a little bit of an empty space that's pretty close to the shape of the state of Kentucky. So our digitization efforts are really filling quite an important hole in terms of biodiversity data. We've been digitizing and databasing specimens using SCAN, or the Symbiota database. And to date, we've digitized over 60,000 specimen records. And our relatively unorthodox approach to digitization includes taking an individual photograph, usually dorsal, of every single specimen, even unidentified material. And this has been really useful to get taxonomic expertise from across the world to folks who aren't coming and visiting the collection to look at specimens. And we've been able to rectify and add identifications to specimens just from folks looking at our photos online and emailing us about them. Now, as I mentioned, our digitization efforts have started within the Coleoptera. And a sub-goal of this effort is basically to come up with regional or state checklists. In our recent checklist of the longhorn beetles of the state of Kentucky, we found that of the 222 species found in the state, 63% of those were new state records, which just echoes the fact that Kentucky has had a very minimal history of having specimen records reflected in the literature. Have you had enough of longhorn beetles? Me too. Let's dig a bit deeper into the other strengths in our holdings. Being a regional collection, our holdings are obviously very Kentucky-centric, as well as the broader Appalachian region. Taxonomically, our collection has 2 main strengths, Cicadelids and Lepidoptera. Our leafhopper strength comes from one of our past curators, Paul Freitag, who described thousands of species of leafhoppers and is still actively helping us curate this portion of the collection in his retirement. Case in point, this wall of Schmidt boxes behind me containing many, many Cicadelid specimens. Our Lepidoptera strength comes from the University of Louisville portion of our collection, which was amassed by Charlie Covell in his career of lepidopterology. I'd be lying to you if I told you I didn't want to tell you about all the other projects we have on the go in the UKIC. We're digitizing the bee specimens of the collection as part of the iDigBees TCN project. We've got a lot of other beetle projects on the go, including the sylphids, the clarids, and the tiger beetles of the state and region. And we're hoping to begin digitizing our Lepidoptera specimens, predominantly the University of Louisville portion of the collection, in coming months. We're also setting up a macrophotography lab, both for stacked images and other entophotographic media. That makes it sound fancy. Well, I could keep going, but I know time is short. Do you have any questions for me? That's just the way I like it. Well, I hope you've enjoyed this brief tour of the UK insect collection. Regionally, a pretty important collection for this part of the world. We're constantly looking to improve the collection through more digitization and having people come and visit and help us in that curation effort. So if you have any questions about our taxonomic holdings or anything else, feel free to get in touch. Well, y'all take care now. Remember what they say down here in Kentucky. Don't look a gift horse flying, it's proboscis.
Just realized I'm wearing the same shirt. This is embarrassing. But happy to take any questions if anyone has them. Yeah, Ainsley.
I don't know if you were here when I was talking about the Tom Barr collection.
I've been meaning to find you, but I haven't yet.
A professor that he proposed his career yet would be able to beetles to us, which from my perspective is good and just, but we should talk about
Definitely. Well, and
any
Eric would be better to answer that question since he's the beetle guy. But I was going to mention to you, all of our carabids are digitized at this point. And so whatever we have in terms of cave stuff would probably be online and photographed. Yeah.
What is the imaging system for it?
So we have a stack shot with a DSLR set up, and that's kind of the in-process unit that we're setting up. Eric does all of his photographs for every specimen with a oh, I'm blanking on the name. A little USB camera. What's it called? Pardon? Dynolite. Yeah, thank you. And the nice thing about that is it's really fast for him to do, you know, an image of every single specimen. So Eric is like a finely tuned machine at going through and digitizing, you know, manually for those specimens. So we've got that set up, and we have a few other kind of microscope cameras and things like that, but that's most of the kind of photographic momentum we have. Ben.
Years ago, when I was doing, I did a catalog of the USNM different collections, and I noticed the least represented state was Kentucky. Like, what's up with this? Why is Kentucky so underrepresented?
That's a great question, and I have no idea. You know, we have a lot of specimens, and there's specimens else in other collections, you know, that have come from collecting efforts here. But I have no clue why. I don't know if anyone else has thoughts. It's just the mystery of the bluegrass. All right. Oh.
During his time at my chart, I had a couple of big inventory projects. One was in Southeast Asia, and one I think was in Colombia. Do you guys have any of that material?
So we don't. We have bycatch from that material, but the Hymenoptera, at least, I think, have followed Mike. Yeah, so we probably do. I don't know how extensive it is, you know, as far as bycatch goes. But again, a lot of that, we haven't gotten to the weevils yet. So that would be one group that's not represented online, but many of the other families would be. Yeah.
Well,
all right, thank you.
Well, thank you to every single one of the presenters in the Collections of the World tour. We had 10 countries represented this year, so I feel it's another successful snapshot of all the wonderful diversity of the Collections network that can join us here online and virtually. I'd also like to thank the other organizers of this symposium, Oliver Keller and Jennifer Hiron, for helping collect all of these tours together. So with that, we have another short break before the next symposium, and we will be resuming here at 1:15. So you have about 14 minutes break, and Christy may have an announcement.